Chapter 22, Prokaryotes, including both domains bacteria and archaea. We'll cover diversity, structure, and metabolism, along with those who cause human disease and heal the environment. The conditions on early Earth led to the evolution of the first prokaryotes. Earth formed approximately 4.54 billion years ago in a mass of hot swirling gases and warm oceans. After a billion years of chemical evolution, along came the first prokaryotic organisms that would have lived in multi-layered mats a few centimeters thick. Today we can study these early arrivers as stromatolites. They're layered in rocks along with carbonate or silicate. Some are still growing in places like San Diego and the warm coastal waters of Australia. Let's take a look at some examples. Here we see a microbial mat which covers 1 meter in diameter. It's growing near a hydrothermal vent on the ocean floor. On the right is a visualization of some microbes with a fluorescent microscope. The first life that evolved on our planet would have looked something similar to these. The picture on the left-hand side shows you some living stromatolites located in Shark Bay, Australia. At one point in time, this is all of the life that would have existed on our planet. On the right-hand side are some fossil stromatolites about 1.5 billion years old. They're found in Glacier Park, which is located in Montana. The ancient atmosphere on early Earth was largely anoxic, so there wasn't much molecular oxygen floating around in gaseous form. So only organisms that were able to grow in anaerobic conditions existed. About 1 billion years after the creation of Earth, phototrophs, which are organisms that convert solar energy into chemical energy, evolved. About 1 billion years after that came cyanobacteria. Cyanobacteria, also known as blue-green algae, evolved to use readily available resources and release oxygen as a waste product. It's thanks to cyanobacteria that our atmospheric oxygen levels increased to a usable amount, forcing the evolution of new organisms that used the molecular oxygen instead of letting its chemical reactive behavior destroy them. Microbes are adaptable, and they live in both moderate and extreme environments. Almost all prokaryotes have a cell wall which allows them to resist changes in pH or dehydration or extreme temperature shifts. Extremophile bacteria, who live in really crazy locations, have grown to prefer them, as there's almost no competition for the resources that they have adapted to thrive on. Examples such as the ones seen here even have special repair mechanisms. Normally, ionizing radiation could destroy all of the DNA in a cell and leave it to die. However, in the instances of some archaea, repair mechanisms exist that allow the DNA to repair the damage, and the cell can continue on growing and dividing as normal. Here are some examples of extremophiles and their preferred conditions. You can see that their conditions for optimal growth are in extreme pHs, temperatures, or concentrations of chemicals that are dangerous to other cells, like salt and sugar. There are some prokaryotes who even live in the Dead Sea, which is a hypersaline basin located between the countries of Jordan and Israel. The Dead Sea has 10 times as much salt as the ocean and contains about 40 times the normal amounts of magnesium, iron, and calcium. Microbiologists typically grow prokaryotes in cultures using a food broth called a medium, which can be liquid or solid material. At the right temperatures and in the right conditions, the bacteria should be able to grow. Should being the key word. 99% of bacteria cannot be grown in cultures. There are simply too many factors for their growth, and we don't know how to control for them yet. It interferes with our ability to study these bacteria and research any potential uses that they may have. Biofilms are present almost everywhere. They clog our pipes and drains and coat the top of our kitchen appliances if we don't clean them correctly. The 5 stages of biofilm development are seen here. During stage 1, the initial attachment, bacteria adhere to the solid surface via weak van der Waals interactions. These are just very light chemical bonds. During stage 2, also known as irreversible attachment, hair-like appendages called pili permanently anchor the bacteria to the surface. In stage 3, which is the first maturation stage, the biofilm grows through normal cell division known as binary fission and actually recruits other bacteria to live with it. An external matrix composed of polysaccharides, or long sugar chains, holds the biofilm together. In stage 4, or maturation 2, The biofilm continues to grow and takes on a more complex 3D shape. In the fifth stage, known as the dispersal stage, the biofilm matrix is actually partially broken down. This allows both some of the bacteria and the gases that they create to escape into the external environment. The bacteria can colonize another surface, and the gases make our houses smell. You could talk about the differences in prokaryotes and eukaryotes for a long time, so let's just touch on their similarities instead. All cells have plasma membranes, cytoplasms, nucleic acids, and ribosomes. So all of the prokaryotes we're going to discuss have those things. All prokaryotes also fit into three basic shapes as well. Cocci are what you see in image A, they're like spheres. B are bacilli, think about Tylenol capsules, and C are corkscrews, known as spirochetes, or spirili. Prokaryotes are unicellular organisms that lack organelles, which are by definition membrane-bound structures. Generally speaking, they don't have chromosomes, but rather a singular circle piece of double-stranded DNA that's centered in a nucleoid region of the cell. Prokaryotic cells stand out because of their cell wall, which can differ in composition depending on which domain you're speaking about. In some cases, there's even yet another layer around the cell wall, known as the capsule. The capsule helps with protection from dehydration and outside attacks. There are pili and flagella sticking out from some cells to assist them in movement and attachment. So even though they don't have organelles, they can be semi-advanced. Both domains of bacteria and archaea contain prokaryotic organisms. You can see these two kingdoms here with the groups or phylums that belong to them listed underneath. We're going to take a look at them individually, starting with domain bacteria, phylum Proteobacteria. There are 5 types of Proteobacteria, and each is actually its own separate phylum, labeled alpha through epsilon. Alphas are photoautotrophic, or symbiotic in nature, and they're thought to be the ancestors of what we know as mitochondria today. Beta bacteria are incredibly diverse. A strand tying most of them together, however, is their basis in the nitrogen cycle. Gamma bacteria are tied to the human gut, for better or for worse. Some are helpful, while others can be quite dangerous. Delta Proteobacteria continue with the diversity. Some are well known for their fruiting bodies, while others draw attention to themselves by reducing sulfate to sulfur. That's that gas that smells like rotten eggs. Epsilon, the last group, can be symbionts or pathogens, and they're found living in hydrothermal vents in places that we used to think of as inhospitable. There are 4 phylum of bacteria that are not considered proteobacteria. The first is the chlamydias, and they're known for their parasitic capabilities and their large lack of peptidoglycan. The spirochetes are spiral in shape and tend to be free-living, but as always, there are exceptions, and some of them cause illness, like the one that causes syphilis in humans. Cyanobacteria, or blue-green algae, are found just about everywhere, and they have the ability to undergo photosynthesis. Oxygen in our atmosphere originally built up because they were pushing it out as a waste product thousands of years before it had any use by any other organism, so the concentration just kept going up. The final group of bacteria we'll look at are the gram-positive bacteria. All bacteria previously mentioned are gram-negative. This is a reference simply to how they react to a specific stain, and we'll talk about that in just a minute. So about those gram-positive bacteria. Generally speaking, they're soil dwellers, and they help to keep the matter cycle going in our planet because they decompose dead things. They have very thick cell walls and no outer membranes. Surprise, surprise, some of these can be pathogenic too. With this slide, we'll begin our overview of archaea. Phylum Archaeota includes organisms that either love methane or salt. Crane and Archaeota are known for their ability to fix carbon, which means they turn it from a useless form to a form that can be passed up the food chain or through the planet's carbon cycle. Many are sulfur dependent and have some degree of thermophilia. Nanarchaeota is a single species that's associated with another species of archaea. It's only been found at the bottom of the ocean and in a hydrothermal vent in Yellowstone. The final phylum is Corearchaeota. We have yet to be able to cultivate this species for further study, but it's been harvested from Yellowstone National Park in a hydrothermal vent. Because it's not been cultivated, we don't know much about it. The cytoplasm of prokaryotic cells contain an incredible amount of dissolved solutes. To keep all that in, plasma membranes surrounding the cells have to be incredibly strong. The structural components in the plasma membrane differ from archaeal to bacterial phospholipids. Knowing their identifying traits can help scientists discern unknown samples from one another. For example, if you ever come across a monolayer of phospholipids that exists in an organism, you know you must be looking at archaea because that is exclusive to their domain. If you look at the fatty acid tails on the phospholipids, you'll also notice an extra branch in the archaeal forms. And there's also a different type of bonding that is attaching the glycerol to the head of the fatty acid chain. Bacteria are divided into two major groups, gram-positive and gram-negative. Both groups have a cell wall that has at least some peptidoglycan in them. In gram-positive bacteria, however, the cell wall is very thick. In gram-negative, it's very thin. In the negative bacteria, the wall is surrounded by an outer membrane that contains lipopolysaccharides and lipoproteins. There are porins, or proteins in the cell membrane, that allow substances to enter and exit the cell membrane in gram-negative bacteria. In gram-positive bacteria, a special acid anchors the cell wall to the cell membrane. This distinction of negative and positive are identifying characteristics that affect the way the bacteria can react to certain external cues, like antibiotics. So we study something's ability to pick up gram staining as a hallmark to know how to treat the infections caused by those types of bacteria. Here's a chart highlighting some of the general differences and similarities between bacteria and archaea. Some of them are quite small. As the organisms are quite small, the differences and similarities are too. As studied in Bio 181, prokaryotes tend to reproduce by binary fission. What we didn't learn in that class were three very cool processes. Transformation is the first, and this is the process in which prokaryotes actually pick up genetic information from the environment around them. In transduction, short pieces of DNA can be shared by viruses to effect change in the bacteria. They end up carrying pieces of the viral infections, can actually carry pieces of one bacteria into the next bacteria along with whatever little piece of viral DNA was infected. This can lead to great genetic changes in few generations. The third process is conjugation, and this is where bacteria directly transfer genetic material from one cell to another. This is how they can share useful qualities, like antibiotic resistance. OpenStax states that cells are essentially well-organized assemblages of macromolecules and water. And that's one of my favorite sentences because it's something very true that isn't often stated so simply. For cells to grow and function, they require nutrients. They have to be able to assemble all of those macromolecules. Nutrients that are required in large quantities are known as macronutrients. They are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. When nutrients are only needed in small amounts, we call them macronutrients. The common ones are iron, boron, chromium, and manganese. Cells are capable of gathering all of the nutrients and energy they need from a variety of places. In this image up top, you see the energy sources for prokaryotes, either sunlight or chemicals. The carbon source is shown on the left-hand side, either organic compounds or CO2. When you cross the two, you get the four major feeding types of prokaryotes. When something gets its carbon from CO2 and its energy from chemicals, we refer to it as a chemoautotroph. If you get your carbon from organic sources and your energy from sunlight, you're considered a photoheterotroph. There are four major types of energy and carbon users in the prokaryotic world. Prokaryotes, using these different energy sources and carbon sources, play a significant role by continuously moving carbon through the biosphere. Down on the bottom, you can see the microbial respiration and decomposition that moves carbon from carbon sinks back into the atmosphere. Take a look at this image and follow the path around. Carbon also plays a key role in the nitrogen cycle. Again, acting as decomposers, some bacteria turn dead or decaying matter into ammonia. Ammonia is not in a usable form for most things on our planet other than nitrifying bacteria. It's capable of turning ammonia back into a nitrate, which is helpful for growth in plants. Take a look at this cycle too to see all of the things that bacteria does with nitrogen, which is a requirement for all eukaryotic cells. Humans have been battling bacteria since long before we knew what they were. Epidemiologists are the individuals currently studying how diseases affect populations. We often hear of epidemics, which are diseases currently occurring in an unusually high number of people. Pandemics is when an epidemic is worldwide. Endemic diseases, however, are diseases that are pretty well consistently present in a population. Worldwide numbers of endemic diseases with vaccines are decreasing. In certain parts of the world, however, like the United States, there are individuals who don't understand vaccine function and are therefore endangering the lives of millions of people by choosing not to vaccinate their children. If the trend continues, we'll hear a lot more about deadly diseases that are 100% preventable, taking the lives of children around the country. There's an incredibly long history of bacterial diseases making a huge impact on our planet. A little bacteria can cause an entire city to fall. The bacteria that causes typhus brought the plague of Athens, which led the city of Athens and Greece to lose the Peloponnesian War. Today, there are between 16 and 33 million cases of typhus that occur annually, resulting in over 200,000 deaths. Carriers of this disease can be asymptomatic, so they have the bacterial infection, but they don't feel ill, so they don't know it. In a famous case in the early 1900s, a cook named Mary Mallon unknowingly spread the disease to over 50 people, 3 of whom died. Other forms of this bacteria regularly cause food poisoning. Here we see 3 images relating back to bacterial diseases. In A, you see the Great Plague of London, which killed an estimated 200,000 people, or about 20% of the city's population. The causative agent is Yersinia pestis. It's a gram-negative, rod-shaped bacteria that belongs to gamma proteobacteria. The disease is transmitted through the bite of an infected flea, which is carried around on the backs of rodents. That's what you see in picture B. Symptoms include swollen lymph nodes, fevers, seizures, and vomiting of blood. Gangrene can also develop, and that's what you see in picture C. Epidemiologists study emerging diseases, which are either in a region for the first time or back in that region after a very long time. 75% of re-emerging diseases are zoonotic in nature, so they're transferred from animals into humans. Lyme disease often, but not always, results in a characteristic bullseye rash as seen in A. The disease is caused by what you see in B, a gram-negative spirochete bacteria from genus Borrelia. The bacteria, you can see the transmission over in C, infects ticks, which in turn affect mice. Deer are actually preferred as a secondary host, and then the ticks might move on to feed on a person. Untreated, the disease causes chronic disorders of the nervous system, eyes, joint, and hearts. The disease is named after Lyme, Connecticut, in which an outbreak occurred in 1995 and subsequently spread. The disease isn't new, though. It's just that we've figured out more information about it and given it a name. We found a mummy that's over 5,000 years old that had Lyme disease then, so it's not a new illness. Now we just know more about it. Another big issue with bacterial diseases is increasing rates of antibiotic resistance. As our environments are washed in antibiotics, we kill off weak bacteria and only the super strong are able to survive. Their descendants, when they do reach humans and cause infection, cannot be easily killed off the antibiotics that they've already overcome. This scanning micrograph shows methicillin-resistant Staphylococcus aureus bacteria, commonly known as MRSA. S. aureus is not always pathogenic, but can cause diseases such as food poisoning and respiratory and skin infections. To avoid an increase in rates of antibiotic resistance, do not ever take antibiotics unless you absolutely need them. Never flush them down the drain in your home, and never share prescriptions with individuals who don't require them. Using antibiotic hand soaps and gels constantly are completely unnecessary, and in most cases, using regular soap and water is perfectly acceptable unless there's an actual concern for bacterial infection. Foodborne disease or food poisoning is an illness resulting from the consumption of pathogenic bacteria, viruses, or other parasites in food. Roughly 76 million people a year in the United States are affected by food poisoning. Some really famous cases are all tied to E. coli, which very easily grows on sprouts. It's important to know that not all bacteria are bad by any means. Foodstuffs like cheese, wine, beer, bread, and yogurt are all made on the dependence of bacteria, thanks to their ability to ferment or release alcohol as they grow. One of the most useful activities of prokaryotes is the fixation of nitrogen. Nitrogen naturally exists as a gas that has to be fixed into a usable form, so prokaryotes can benefit from it to build proteins and nucleotides. This process, known as biological nitrogen fixation, is carried out by organisms like soil bacteria and cyanobacteria. Without nitrogen-fixing bacteria, life as we know it would end. Another wonderful function of prokaryotes is bioremediation. This is the removal of dangerous materials in the environment by prokaryotes. They can use various harmful substances via natural metabolic activity and clean up environments. Bioremediation is being explored continuously, especially as we crisscross our planet with things like oil pipes that ruin water sources. With that, we end chapter 22. Go back and read the chapter, take notes, but make sure you do so in your own words. Do the practice problems in OpenStax, do the activities in Learner, do the practice problems in Learner, and then finish your homework assignments. Remember, the more things you do to physically test yourself on what you've heard and read, the better you're going to do when it comes to taking an actual test.